Hi everyone. Thank you for coming to hear about my poster. My name is Reiman, and I'm a research assistant professor at the Toyota Technological Institute at Chicago, and today I'm excited to talk to you about some work involving a deep learning approach for modeling longitudinal microbiome data. For those that may not be familiar with the microbiome, it consists of a community of microscopic organisms such as bacteria, fungi, and viruses, and this community is believed to functionally interact with its environment. When we think about the human microbiome, we know it can be affected by multiple host characteristics, behaviors, and external stimuli that can perturb this community. In particular, the human gut microbiome has been shown to impact host development, normal metabolic processes, as well as the pathogenesis of various diseases. Based on these discoveries, the engineering of the gut microbiome for the treatment of such diseases has become an exciting new direction in medical science. And uncovering the nature of how to precisely control a patient's microbiome requires accurate modeling of the dynamics of the microbiome community under varying conditions. However, the modeling of longitudinal microbiome data faces many challenges due to the inherent noise of microbiome data. Therefore, the development of robust and accurate modeling techniques for longitudinal microbiome data will empower the identification of microbiome-targeted therapies. To address this challenge, we have developed a framework which we have named DiRLaM, which combines an autoencoder neural network with a supervised neural network for the modeling of longitudinal microbiome data. We use the autoencoder first to perform dimension reduction, to learn a reduced latent space that captures the intrinsic structure of the data. Then, in the reduced latent space, we first perform interpolation to smooth the longitudinal data, and then we integrate host characteristics as well as any external factors which may perturb the microbiome dynamics, and use the latent representation of the microbiome community at time t. We try to predict the new latent representation at time t plus 1, which can then be decoded back out into the microbiome feature space. Using these two trained networks, DiRLaM can then group microbes based on similar dynamic patterns and identify which factors and characteristics are significant in modulating the dynamics of the microbiome community structure, allowing for the construction of a module-based interaction network. One novel piece of DiRLaM is how the autoencoder is trained. During training, interpolation between two samples is performed using a random mixing coefficient, and a novel beta diversity regularization is applied to encourage that the total distance between the two microbial community inputs is equal to the sum of the distances between the interpolated microbiome community and each input, essentially causing the interpolation to happen in the shortest possible path with respect to how we characterize distance in the microbiome data. To evaluate DiRLaM, we simulated longitudinal microbiome data by generating synthetic data using the R tool Microbiome DA-Sim. We created longitudinal profiles for 100 subjects, each with 120 evenly spaced time points and 100 microbial features. We then created alterations by randomly applying 3 discrete events during the duration of the longitudinal data, where the presence of each event alters the dynamics of 10 microbial features, some of which are shared across events. Since real-world microbiome data is never this densely sampled nor clean, we created corrupted data sets by dropping time points ranging from 50 to 90%, as well as by masking a subset of features at each time point ranging from 10 to 50%. We evaluated DiRLaM using 10-fold cross-validation, where models were trained on the corrupted data and held-up predictions were evaluated using the uncorrupted. First, we looked at the effect of the beta diversity regularization strength, and we observed that although stronger regularizations were beneficial at higher corruption, they were also detrimental in data with less corruption. Based on this and the fact that we can never know how much noise is truly in the data, we selected a strength of 2 for further benchmarks. The next thing we evaluated was DiRLaM's interpolation ability, and we compared DiRLaM to a standard B-spline interpolation, a similar linear interpolation within a reduced space defined by principal component analysis, and a standard autoencoder, meaning that there was no beta diversity regularization. We observed that all methods performed comparable in lower corruption settings. However, DiRLaM was more robust compared to B-spline and PCA interpolations as the level of noise increased in the data, highlighting its ability to capture the true signal of the data and create smooth interpolations. Lastly, we evaluated DiRLaM's ability to predict subsequent time points. We first compared DiRLaM to a standard approach of using a dynamic Bayesian network, or DBN, on B-spline smooth data. Then, based on our observation that DiRLaM improved interpolation, we trained a DBN using DiRLaM's interpolated values. We then considered that DBNs suffer at larger feature spaces and evaluated how DBNs perform using DiRLaM's reduced interpolation within that latent space. What we noticed is that within each change, the DBN models improved, showing that dimension reduction can even facilitate the training of debian models. However, despite the improvements in debian models, DiRLaM was still able to outperform DBNs using its neural network in each instance. Now we're going to turn to a real-world biological data to demonstrate what DiRLaM can learn. We will be using a longitudinal microbiome dataset from a study by La Rosa and others that looked at 58 infant guts over about 26 time points, giving a total of 922 samples and 29 microbial features. Host characteristics included gestational age, post-conceptional age, mode of birth, and biological sex. External factors included the amount of breast milk consumed, amount of antibiotics used, and room ID. Within this dataset, DiRLaM identified 4 significant factors: antibiotics, amount of breast milk consumed, mode of birth, and biological sex. We can then take these factors and create our module-based interaction network. DiRLaM was able to learn that increasing breast milk consumption positively affected the dynamics of gamma proteobacteria, which has been observed in previous studies as well. Additionally, infants who underwent vaginal delivery had an increase in actinobacteria, bacilli, and clostridia, each of which has been shown in other studies to be associated with vaginal delivery. In conclusion, we presented DiRLaM, a combination of an autoencoder and supervised neural network for modeling microbiome dynamics. DiRLaM applies a novel beta diversity regularization to help create smoother interpolations, and we showed that DiRLaM improves upon current interpolation and dynamic modeling techniques. Additionally, DiRLaM was able to learn significant and informative factors in real-world longitudinal microbiome data. I would like to thank Dr. Yang Dai and our lab, as well as additional collaborators. I would further like to thank you for watching, and if you have any questions, please feel free to reach out, as I would love to discuss my work.